Welcome to this Academy for Teachers masterclass on virus evolution with Paul Turner. I'm Emily Holliday, and I have been teaching science for the past 6 years at West End Secondary School. I love what I do because for me, teaching is very creative. I love having an idea about how to engage students with a topic, trying it out in the classroom, and getting the chance to improve my approach the next day. I'm a proud fellow of the Academy for Teachers, an organization that gives me energy just when I need it. I have the honor of introducing Paul Turner. Paul is the Rachel Carson Professor of Ecology and Evolutionary Biology at Yale University. He uses an interdisciplinary approach to examine how viruses adapt to overcome new challenges. His laboratory uses an interdisciplinary approach to investigate these processes, employing techniques from microbiology, population genetics, genomics, molecular biology, and mathematical modeling. I'm excited to take this class because I love to hear directly from scientists about their work. But Paul is not the only star of this masterclass. It is also my honor to introduce the 24 teachers here today who were selected from many applicants. This is an impressive group with advanced degrees in biology, education, life sciences, science journalism, and law. They teach grades 4 through 12 and early college in public schools, charter schools, and independent schools. We teach them really interesting subjects like Introduction to History and Geography, City and Sustainability, Molecular Cell Biology, and Anatomy and Physiology, to name a few. We teach between 40 and 240 kids. The number of children who know our names is 2,929. That's this year alone. So your real audience today, Paul, are those 2,929 kids, because every teacher here will, in some way, share with them what we learned from you. The ripple effect of a masterclass is huge. Thank you, Professor Turner, for being here. Thank you to my fellow teachers for teaching. And with that, let the masterclass begin. Paul, take it away.
Wow, that was a terrific introduction. Thank you very much. Thank you all for the terrific work that you do. I come from a long line of teachers. My mom was a teacher. She's no longer living, but she taught public school in Syracuse, New York for several decades. And my younger sister, she still lives in upstate New York, and she teaches in public school in Syracuse as well. That's just a couple of my close relatives. I won't get into the other ones who do teaching, but indeed, your work, frankly, is more important than mine, I would say. So it's wonderful to be with you and in your presence today. I appreciate that very kind introduction. So I thought what I would do, since I think I had a lot of ability to craft this kind of however I wanted, is I want to hold forth with some slides for about 20 minutes maybe, and then we'll take a break and discuss that content on virus evolution, and then I'll show some more slides and present some more work more on the applied uses of viruses, which requires evolution thinking, but it's more in the realm of virotherapy. And I think we have until 5:15, is that right? More or less? Okay, all right. So great, should I just take it away, share screen and all that stuff? All right, fingers crossed that this works. Let's see that. And can you still see that and hear me?
Yep.
Awesome.
All good.
Okay. So pleasure to be here. And the topic to begin with is virus evolution. And oh, by the way, reading your credentials, you know, the write-up that I was given for you all, you are a pretty amazing group. And it sounds like you, many of you especially, have some deep knowledge of this stuff already. So I apologize if some of it is a little redundant for things that you've heard before. Okay, so first of all, where do viruses come from? I'm an evolutionary biologist, and I don't do as much of this kind of work on patterns of evolution. That's what's shown here. This is a phylogenetic tree. It's obviously one way of depicting the tree of life. And life is in quotes. It's a little uncertain what life really should be defined to be. We discussed that a little bit in the breakout room that I attended. You are there in that pink portion of this diagram among the eukaryotes, and your close relatives as the one of the other three domains are the archaea, single-celled organisms. And most of this particular depiction is showing the biodiversity among bacteria. Can you see that cursor? It's okay? Okay, I hope so. There are no viruses on this tree of life. And the reason is that there are genes, especially ribosomal genes, that you can sequence and find commonality for all of these cellular organisms and put them in the same tree based on their relatedness of ribosomal genes, but you can't do that for viruses because they have no metabolism and therefore no ribosomal genes. And there are a lot of ideas about where viruses might have come from. This one I find the most compelling, but I should state clearly it's not necessarily the most correct. And there could be more than one origin of viruses, but this is one that I love, basically because it's impossible to even test it. So it kind of caters to my interest in science fiction, maybe. But viruses might have predated the evolution of cellular life. And sometimes when you hear people talk about the deep evolution of cellular life, they talk about LUCA, which stands for last unique common ancestor. So the idea is maybe way back, even before cellular life arose, you had an ancient virosphere. It sort of followed after an RNA world, for example. And what you might see to the current day here, they're trying to depict in this article, how the modern virosphere is specific to these three domains, the bacteria, the archaea, and the eukaryotes. But they might be viruses that were around for literally billions of years. And this arrow here is talking about how among single-celled organisms, there might be an easier path for viruses to transit from one domain to another one. So the point is, I don't know that this is correct. I find it really compelling. I said that earlier. And it's kind of one of these absence of evidence feeds into the argument things that if you take cellular life and you fully sequence it and you look at its genetic content and you do the same thing for viruses, sometimes you don't find an overlap. And that means it may not be the case that viruses are just some rudimentary form of cellular life that splits off all the time and so on. It could be that it's ancient and you don't find homology because they pre-exist cellular life. Not sure. Those patterns that I just showed in the tree of life, this is more of my wheelhouse. I'm an evolutionary process person. I study how processes lead to those patterns. And certainly one of the most impactful discoveries ever in biology, if not all of science, is Darwin's articulation of natural selection. So he was not really the first person to put forward an idea of how life evolves, but he was the one who got it most correct. And in his most famous book, Origin of Species, I mean, he lays it out for natural selection to go as following. If you have individuals in a population that vary and that variation is heritable, then some of those variants are going to be advantaged in survival and reproduction. And if that's the case, then the population is going to evolve to reflect the successful ones. Now, this leads to some very important consequences, especially trait evolution. So you get from this adaptations. The better survival and reproduction is due to traits that some variants have and others don't have those same traits. And we call those adaptations as a very specific definition. And one more thing I could say about Darwin's thinking and his contribution here is that indeed it could be a universal law in biology, for which we have very few universal laws in biology, I would argue. And by this, I mean that if you have this happening in a non-carbon based life form, let's say in the atmosphere of Venus, if you have such individuals and populations there, could we predict that if this same thing holds, that those populations are made up of individuals who vary and that variation is heritable in some way, are you going to get the same sort of a process playing out, even though it would be life that evolved completely independently of the life on this planet? So that, again, another compelling idea. But I will say that there's a lot of physics envy in biology, that we don't have very many universal rules, and this could be one. So I'm now going to talk a little bit about one virus system specifically, and it's only because it's a convenient one to talk about in the sense that it is probably the biological system we know the most about. That might change now that we're dealing with the pandemic and SARS coronavirus too, but the point is in HIV, we've been grappling with this epidemic for a long period of time. And when HIV first emerged as a problem, there was a wonder drug that came not that long after it. It's called AZT, azithromycin, and this was approved in the late 80s. But the failure of it it's basically evolution by natural selection that caused this wonder drug to fail. And I want to go through the steps of that because I think it is a nice example of how viruses are always evolving faster than their hosts. They tend to do this, and this could be problematic for their host to keep pace. So what does AZT do? This is a very busy diagram, but it's just a reminder that if HIV is encountering a cell that it can enter, the CD4 receptors there, then it gets in, and there's a very crucial step that occurs. It's an RNA virus. So there has to be this step for a retrovirus to create a DNA equivalent within the cell that gets incorporated into the genome of the cell that ultimately instructs the cell to make virus particles that are RNA. So this is a transition from an RNA to a DNA state, and that happens through reverse transcriptase. So here is that enzyme. If it's trucking along here and encountering an RNA template and making a DNA version of it, it's gathering up these nucleotides within the cytoplasm to do so. And the thing is, can you get this step to be stopped? So if you have a drug that stops this process from occurring, then it would seem to be a wonder drug because it is a key step that the virus has to undergo to go from an RNA to a DNA version before it goes back into instructing the cell that RNA viruses should be made. So what is AZT? It's basically AZT triphosphate here that has the wrong group. It has an N3 group rather than an OH group. And when this transcriptase enzyme is fooled into bringing it into this chain, thinking it's ordinary thymidine, then it's got a problem. It should stutter out and stop because the OH group is missing. So AZT fools this enzyme by mimicking the normal nucleotide thymidine. And it lacks this attachment site for the next nucleotide in the chain. Okay, that's all well and good, except for the following problem. Suppose you can have a resistant form of this reverse transcriptase that's coded by the virus such that it does not recognize picking up this imposter, right? It has a change in the active site so that it won't do this, even though you want it to. And a virus with a mutated active site is obviously going to be selectively favored within the host because it's going to be able to replicate. A reminder that if you have these mutations occurring, any mutations are raw material for natural selection. And mutation rate is very high in HIV. As we're reminded by this is a review article by Siobhan Duffy and some others Siobhan was one of my PhD students. HIV does have a very high mutation rate, and this is typical of most RNA viruses. This is kind of a tricky graph, but if you consider genome size along this y-axis and mutation rate, you've got slower rates of mutation to the right. And what it's showing is that if you just catalog mutation rates in viruses of different type, of different nucleic acid type, then HIV is nested right in here among the retroviruses, which are these yellow triangles. So they don't have the fastest mutation rates, but they certainly have faster mutation rates than a lot of other viruses. And now the take-home message of what I'm getting to here is that the high mutation rate in HIV, as demonstrated in this diagram that I'll walk you through, natural selection is what's causing AZT to fail in every last person who was given this wonder drug. So what's happening here? This is depicting an expanding virus population within the human body. And let's start at this end where the original variant that's in there is the wild type pathogen, and it is susceptible to AZT. In other words, it should not be capable of replicating if AZT is in the system. So the errors in reverse transcription, though, in this virus population are going to lead up to mutations and different mutational variants just being there. And that just happens because they have a high mutation rate. So now through time, eventually it gets to this step where you see these circles with the red line through them. And that is when the physician has put AZT into the system thinking it's going to stop this process from going forward. Well, unfortunately, what's already happened in that virus population is that there are already partly resistant variants of HIV to AZT and also highly resistant ones in blue. And interestingly, they're there even though AZT was not in the system at the time. They're just there because there's a high mutation rate in the system. And if you work your way across this busy cartoon, it begins with individuals are varying. Variation is heritable. Some of those variants are advantaged once AZT comes into the system, And what happens is ultimately the population evolves to be reflecting the most successful variants, and that's evolution by natural selection. So unfortunately, this happened in patient after patient after patient. When the AZT was introduced, eventually it failed, and it turned out to not be a wonder drug. So we still grapple with HIV and using combinations of drugs to keep people alive who are HIV infected and preventing them from converting into AIDS patients where their immune system collapses on them due to the destruction of immune cells by the virus. And depicted here are lots of examples. Here to the left are some pretty familiar ones. Coronavirus is shutting down the world right now, still, SARS coronavirus 2. And a lot of my work deals with this problem is how can you better predict what's going to emerge next? This is a huge problem that is now elevated to a very big problem because we want to be better prepared in case this happens in the future. And it will happen in the future is the point. We just want to be as best prepared for it as possible. And there's some general questions I could throw at this just to illustrate the kind of things that are on my mind. Why is it that some viruses are good at infecting multiple hosts? So if you have a virus that is in a bat naturally, but could spill over into humans, then in a sense, it's got an innate ability to be generalist in its ability to infect host types. And that might be at the heart of what's driving emergence. If you've got some viruses that are out there that naturally infect multiple hosts, maybe they're poised to jump into a new host such as humans. That's just one idea. Let's talk about HIV a little bit more. And we grapple with where do these emerging viruses come from in the first place? And we know a lot of information on HIV, and the story goes like this. If you look at actually the diversity of HIV that's out there, especially in HIV-1, it falls into these clusters. And it's quite obvious that these different clusters, if you look at the sequences of viruses that are in close relatives and distant relatives among the primates, so chimpanzees, and maybe in some monkey species, you can find that the HIV samples from humans are very closely related to some of these samples coming from non-human primates. So what certainly has happened, and I'm happy to talk about the details of it, is that on separate occasions, SIV, simian immunodeficiency virus, has jumped into humans multiple times. And that is the best explanation why you have these clusters of HIV that are closely related and more distantly related to other HIV. So a lot of this work came from Beatrice Hahn. I think she's at University of Pennsylvania. But her work is just amazing to look at these gene sequences, especially viral surface proteins, and try and make sense of this. A little bit more on this is, if you have ever seen phylogenetics, this is kind of a funny tree. It's called an unrooted tree. But basically, there was this cool study published in 2000 that I'll walk through it in a couple of slides because it's one of my favorite papers that I didn't produce, but I'm very impressed by it. So you can do this kind of unrooted tree if you take samples of HIV from sick people. And this particular analysis had 159 of them, and they fully sequenced all of those viruses. You don't know the structure of this tree. You can root it at some inferred common ancestor, but the distance from the tip of each of these 159 samples down to the common ancestor, that's a distance genetically for which they have diverged from that common ancestor. And you can lay that out in a graph like this. The divergence from the common ancestor is on this axis, and the year at which these were sampled is along the x-axis. And what this does is allow you to infer, well, ultimately it sets up this very nice example of how across time, there's kind of like a clock-like way that these viruses are evolving to become more diverged from the common ancestor if they are, time has elapsed since they have evolved from that common ancestor, depending on when they ended up in the clinic and in this collection. But the point is, you can get this relationship, and this line is shown here, and you can do something else with it. So now you can't see all the details in that prior graph, but it's the same one I just showed you. And you could take that line and you could extrapolate down to hit the x-axis at a very special point in time. And that special point in time would be our prediction, or the prediction of these researchers, and when exactly did HIV-1 first come into the human population. And this is the thing that blows my mind. And I think the students, when I present this to them in class, they're like, wait, what? HIV-1 might have emerged in humans in the 1930s? And it's indicative of it can take a very long time to figure out that an emerging virus is causing sickness in humans. It has to be doing it at a high enough rate that you make sense of it all. And literally that might have happened over, nearly a half a century for us to figure out what was going on. Now, this is an inaccurate analysis, right? Because you're extrapolating back, and that's why you have this very big breadth here, this 95% statistical confidence around that estimate. But still, it's not a bad estimate. This isn't that many years separating it from the low versus the high estimation of this. And somewhere between 1915 and 1941, it's very likely that HIV-1 first emerged in humans, even though we didn't really figure this out until enough clusters of cases were happening in cities of large size. And that didn't happen until the 1980s. So it's pretty cool. And I guess the punchline is there are many things going on now, aside from the pandemic, that are the future emerging virus problems next year or the year after, and we just don't know it yet. Okay, that's the sobering news. So the time at which some virus and host have been in one another's presence and have evolved or co-evolved and had an association is sometimes clear, sometimes not. So I just went through an example of a relatively recently emerging virus, HIV, but we can find other viruses, such as in primates, humans and other primates, that are quite ancient. And we know this to be the case because if you have these nice phylogenies here on the right of some species, in this case it's humans and non-human primates, you can sometimes find a mirror phylogeny for, in this case, viruses that are related to one another, but clearly the mirror phylogeny says that they're highly specific to these species. And what is suggested by this is the relationship between cytomegaloviruses and humans and other primates is probably so ancient that it's way back when primates first became primates and started to diverge. And as they diverged, they carried along with them these cytomegaloviruses that were specific to them. So that's just a bit of how you can have ancient relationships. And now we jump again to an example from the present. It's unclear sometimes, how recent have events occurred unless we have great evidence. In the case of MERS, we have great evidence. So this was discovered in Saudi Arabia in 2012. Very nasty fatality rate. It's hard to tell people that the current pandemic could be worse. This is a related virus to coronavirus, SARS coronavirus 2. This would be much worse. If MERS really emerged on a global scale and 1 in 3 people were dying from it, that would be horrific, right? So it could be worse, it's sad to say. Where did this come from? Again, we turn to phylogenetic evidence. Human MERS coronaviruses seem to be very closely related to those in camels. So this story emerged, I'm sorry not to use the word too many times, but the story is that people who were tending domesticated camels came into contact with the viruses that were shed by the camels and they jumped into humans, and that was MERS coronavirus. And from there, we can find that the next closest relatives to these viruses in camels are bats. And by now, I'm sure you've heard about the role of bats potentially as the ultimate reservoir source for many types of viruses that are emerging in other mammals besides humans, and then sometimes making it all the way to humans. So this is the kind of evidence that tells you that. Now, if you go back prehistory for humans, you could see some ancient virus diseases and the evidence for them, but really it's sketchy. It's difficult to know when did those first emerge? When did they first come into human populations? And yet from hieroglyphics, ancient remains, say in ancient Egypt, you can find here a man who's depicted who has a deformed leg. And this is a characteristically kind of deformed leg if somebody has a poliovirus infection during childhood. And in the mummified remains of Pharaoh Siptah, it's quite clear that he has a club foot as well. So he suffered with poliovirus way back circa 1200 BC. Pharaoh Ramses V, a little more recent here. So a more recent problem that we have eradicated is smallpox virus. So this is actually a black and white photo of somebody who has smallpox lesions, which is very characteristic of this often deadly disease that used to circulate in humans until we eradicated it through vaccine campaign. But if you look at Pharaoh Ramses V, you can't quite make it out in this image, but he has the scars of smallpox infection that he suffered, and it probably killed him ultimately. So we look at this ancient evidence, sometimes in mummified remains, to try and figure out just how ancient are some of the viruses that we still grapple with today. So I'll stop this section in a couple of moments, but now I want to step back from it. And everything that I just told you is basically to impress upon you that viruses have an amazing capacity to evolve. They might have been around even before cellular life and managed to evolve successfully so that they're here to the present day. They certainly cause a lot of pain and woe in humans, and they have an ability to spread rapidly, as we're all facing this right now. And this happens again and again and again. So, okay, that aside, I find it remarkable that viruses are as successful as they are. And this is a big part of my research program. These are different, what you'd call biological levels. So at a very base level of molecular and cellular biology, viruses can infect cells if the right proteins are there to bind to. But viruses don't have, like dedicated movement. So they can't move into the right environment to find those cells. And essentially they're just kind of often wandering around hoping to bombard the right cell type. And to me, that seems like a very difficult way to make it on this planet. And yet they are the most numerous thing on the planet. And as they're moving between different tissues in our bodies or moving between different single-celled organisms of different types, that might fail for their ability to replicate in those cells. And you'd think that they would have a very difficult time with it. They face host immunity, other aspects of host biology that are innate and immune defenses. You'd think that this would be a very tough road. And sometimes they have as few as literally 5 genes, and they're able to grow within the midgut of this mosquito. And as it feeds for a blood meal on a mammal like a human, that virus will get into your body and replicate in you as well. And maybe I need to remind you, I probably don't have to, that this is a very distant cousin of yours. This is a eukaryote, but it is very, very distantly related to you. So I'm fascinated by the ability of viruses to do that with only a handful of genes. And the last is at the level of ecosystems, we are grappling with climate change, global warming, and this is affecting all species on the planet, including the smallest ones in size, and those would be viruses. So we study all of these aspects of what I think are the difficulties for viruses to evolve. How do they make it through all of this? And ultimately, especially with climate change and global warming, the sad truth is organisms like polar bears are probably fated for extinction. So the particular ecology that they live in is large expanses of ice. And what they do is look around those ice expanses for prey, but they have to then swim across open water from ice expanse to ice expanse. And the ice is shrinking and disappearing. So that's why I showed this poor polar bear on, what's symbolizing its shrinking habitat. They cannot swim long distances and they will die if the ice shrinks too much. So they're just not going to make it. And that's why the uncertainty of how the environment changes, changing rapidly and unpredictably, organisms like this that can evolve quickly are probably not going to last. So the punchline here is that even though viruses have an amazing adaptive capacity, they are not so good that they can do everything. And it's unclear whether they could make it in an unpredictable world as well. So I'm going to stop sharing for a moment. And hopefully that was clear enough. I wanted to pause and see if anybody had questions, which actually might be in the chat. Okay. Does anybody want to ask a question that they raised in the chat in real time?
Hi.
Hi.
I was asking about, if you said viruses, RNA viruses mutate at a relatively quick rate, how would a vaccine for the COVID virus be effective?
Right. So here we are, all the evidence suggests that we are lucky in a sense. Our main concern with these vaccines are whether they're going to be providing long-lasting immunity for some appreciable amount of time. And it seems like we are not set up for a scenario where we have to chase an evolving moving target. So coronaviruses can mutate and evolve. They don't seem to do it very quickly. And that's, whew, right? I mean, that means we can produce these vaccines, put them into people, get them safe through mimicking the natural immune response, and not have to worry, maybe not even have to worry on the cycle of every year like we worry about influenza. Flu viruses do evolve quickly. Coronaviruses do not, thankfully. So I hope that answers the question. Anybody want to ask another one in real time?
I guess I have a question related to that. What if when we give the vaccine, if that imposes, I mean, is there any way to change the rate of evolution, or is that kind of like we know it's set?
Okay, I'll try to answer this succinctly because this is really intriguing. You actually don't hear much about it in the news because I think it's a complex topic. So let's say if we were very on board with social distancing and other ways of preventing rapid movement of SARS coronavirus 2 from individual to individual, you're essentially, you're altering its transmission success. And what theory predicts, and what we have actually seen evolve in pathogens, is that if you prevent pathogens from reaching the next host quickly, their current host matters more to them. And it tends to be that they'll evolve a lower state of virulence in that current host because it is more precious to them. So merely the things that we are doing now to prevent transmission from happening rapidly, if there is selection that imposes on this virus to change, it should be causing it to change so that it does less harm to its current host. That's what the theory predicts. That's the good news. The downside is the interaction of this virus with human to human across age groups, or just these mystifying ways of does it make somebody sick or not, it breaks that theoretical prediction. It makes it a more difficult one because that prediction is based on if it needs that host, it replicates in that host, and it has a certain amount of virulence that it impacts in that host system. And if you have this huge degree of asymptomatic all the way to causing death, then it's uncertain how that kind of feedback is going to happen through selection. And basically, your question, though, was about vaccines. So if you vaccinate and prevent people from being susceptible because they're vaccinated, it should cause the same thing that I just said, theoretically, where you will make the current host more precious to the virus, and it will not be as virulent in that host. These are predictions because there's a vast number of other predictions about vaccine pressure and how it changes pathogens, and a lot of uncertainty and actually debate in pathogen research. And the last thing I'll say, Tracy, is I really like your background there. Those look like phages with party hats. That's awesome.
My student made them. I just have to credit him. He said it was okay to share them.
It's good stuff. Anybody else have a question?
I have a question.
Please.
Oh, sorry.
No, go
I
ahead.
have a question that I put into the chat.
Good.
I hope I'm not skipping. It looks like, oh, actually, there are a few people who are ahead of me.
Yeah, I know. Maybe I should be going through these one by one. Should I do that, folks?
That might be
Maybe I
fair.
should. Okay. Oh, wow. I love the fairness. This is good. Okay. So let's see. If COVID is an RNA virus, how will vaccine be effective? Okay, I think I answered that. Vaccine is using mRNA for spike protein. Immune response not ever having pathogens. That's my understanding. Yes. So essentially, any vaccine is trying to mimic the pathogen enough that it fools your immune system into thinking it's the pathogen, and you are not hurt in that process, but you build up the immunity. So that's essentially your understanding is correct. Okay, we talked about influenza evolution, mutation. Yep. Yeah. So essentially, I mean, this could happen. Everything that I've said is true, but you've heard now for months about this particular pathogen. It breaks the rules of pathogens that we've known of in the past interacting with humans. And that's what really makes it a terrifically difficult scientific problem when you're really dealing with something. This is essentially one of the perfect ways for a virus or another pathogen to emerge in a host and have the host be just like, what is going on? Because if you have uncertainty for interaction of a pathogen for its effects on a host, it makes it very difficult to predict what's going to happen next. And unfortunately, you just sort of learn through your mistakes along the way. Yeah, it has a different sort of a mutation compared to other virus types. I kind of said that. Okay. Proofreading. Okay, yeah, this sounds like these are about proofreading and mutation rate. I think I've kind of covered this. How is this possible? Oh, uh-huh, yeah. Takes time for adaptation. But so it used to be the thought that, okay, I can study adaptation in the laboratory, in fruit flies, and something with very short generation time, and I get impactful results in a short period of time in my experiments. That is true, but I think we're getting better as scientists, as evolutionary biologists, looking at the signatures of evolutionary change, especially in genome sequences, and getting stronger conclusions from those data to tell us whether or not a system is evolving quickly or slowly. So it's a bit of a digression, but even in humans, where people talk about, oh, we have medicines, we have lots of ways of keeping people alive that frankly, if selection was acting, they would be removed from the population. And those alleles and those mutations would be kind of pushed out. But it's a reminder that if you're going to force out like a disease allele in a population like ours, it takes a long, long period of time to eradicate it anyway. So there's not that much that we're doing with medical technology that really works against the action of selection to push things out. Eventually that can happen. I kind of muddied that description. Maybe my main point is that evolutionary change is detectable even in humans. And a lot of people, they're like, what, really? Yeah, sure. I mean, you could see it not only, is our diet improving in many countries, not all, over time, leading to physiologically larger people, right? But there are other ways that you could look within those populations and still see traits in men and women that seem to be changing over time. Okay, so there's plenty of evidence for that. Where is that? Larger scale. Huh. I think MERS is not very transmissible. I'm not an expert on MERS, but there I think we got very lucky that we see some of this in coronaviruses. So just one quick anecdote. I went to a meeting in, I think it was late January in Washington, D.C. It was like a emerging biothreats meeting, and Tony Fauci was there. And we were in a room, literally like thousands of scientists. And he starts showing these pictures of, boy, look at these data in Asia and how quickly this spread is occurring. And he was not making light of it at all. But when he said things like, we've been so focused on virus emergence problems that we should have been more appreciative of coronaviruses. We had SARS coronavirus 1, we had MERS by that time, and he's reminding us of how we're going to be grappling with SARS coronavirus 2. Literally a month after that, the world shut down, right? But in that room, people were kind of like, oh, yeah, we really missed it. Boy, we screwed that up. Yeah, I guess we'll learn from our mistakes. And his main message is that we should have respected coronaviruses at SARS coronavirus 1. We should have been more intensively studying them and how to produce vaccines against them. And the wake-up call should have been MERS. And then we kind of still like, those crazy coronaviruses. I don't know what they'll do next, but we better get back to making that flu vaccine. And it's pretty amazing how things can just turn on a dime that way. But I'll never forget that meeting, of course, because Tony's in the news all the time. We had a whole room full of people, nobody with masks on, and the virus was already in the United States at that time, right? So it was pretty sobering. I'm spending a lot of time on these questions. I'm going to take one more. Yeah, bats. Bats seem to have a physiology that tolerates viruses living within them. And not exactly clear why, but some people think it's because many bats, their physiology just drops when they're roosting. Their body temperature goes way down. Whereas when they're active and they're flying around, they have some of the higher metabolisms in the mammalian world. They're kind of like rodents. So they have this huge dynamic range in their physiology. And there's something about that seems to be correlating with their ability to withstand viruses being in them. And so therefore they're sort of flying around as these bags full of viruses with high diversity because their physiology allows it to be so. And that is why they may be serving as reservoirs. So okay if I go back to the slides? Is it okay? All right, let me close this out. I'll share again. Go here. Okay. Oops. All right. So let's talk a little bit about the good that viruses can do. And I'll start first with ecosystems because that's kind of where I stopped at the first half, is that there may be ecosystem level changes that would be difficult for viruses to overcome. But now let's think about just how ecosystems function and what role viruses could be playing in them. Here's a reminder that the blue-green algae, these are not really plants, these are bacteria, these are cyanobacteria. They were the first photosynthetic organisms to arise on the planet. And had they not evolved, then the level of oxygen in the atmosphere would not have increased so much, making it possible for large-bodied organisms like us to evolve later on. So we have the cyanobacteria to thank for that. And cyanophages are estimated to be some of the most prevalent viruses on this planet. They live in the oceans as well, and they kill cyanobacteria. So what they are doing, here's a picture of cyanophages, and I forgot to say these are cyanobacteria here, and this color image is showing you kind of rates of photosynthesis in the ocean. But these cyanophages are so specific in killing cyanobacteria that arguably they are kind of like a predator that is regulating the size of their prey, cyanobacteria. And it's kind of keeping it at a relatively constant level in marine food webs. So the consequence of this is that the viruses, these cyanophages, are kind of regulating the amount of photosynthesis that happens in the oceans. And to me, I think that that's a good thing, because bacteria, if they're greatly fluctuating in their abilities to photosynthesize in the ocean, they're not going through these huge boom and bust cycles because cyanophages are keeping them relatively constant. And therefore that is keeping the atmosphere relatively constant for the amount of oxygen that it presents. Another thing that these viruses will do in the ocean is that they're involved in what's called a viral shunt. And that, another way to say that is that they're basically killing these cells at such high amounts that it is allowing carbon to go back in to geochemical cycling, making it available for other things. So viruses are playing this huge role in the ocean, and it's unclear whether they play kind of the same role in terrestrial environments, but my research is starting to look at that. Classically, it's shown to happen in the oceans. Now, a lot of viruses, you hear about the bad things, but they can be mutualistic with their hosts. An example from a review article by a friend of mine, Marilyn Rusink, is she's very good at studying virus mutualisms. And she's found that, for example, these grasses that are growing in very hot temperature environments in places like Yellowstone, that these plants have within them fungi that allows them to survive high temperatures. But the fungi are allowed to survive the high temperatures because they are infected by a kind of a virus that brings this trait to the fungus and therefore allows the plant to survive. So that's like a three-way interaction that explains why these plants can grow in environments that you wouldn't expect them to. A little more complicated is the story of the parasitoid wasp. So these sorts of wasps are among the most speciose eukaryotes. There are maybe 30,000 species of these wasps, and they are uniquely associated with a kind of virus that's in their genome, and it acts as a mutualist. And the reason for this is that these parasitoid wasps are very specific to some insect larvae, a caterpillar of a particular host species of insect. And what she'll do is she'll come in here and she'll oviposit or lay her eggs in the body of this caterpillar. At the same time, she's releasing these virus particles called polydnaviruses, which is a way of pronouncing this. It's poly-DNA virus. So these are within the genomes of the wasps, released at the time that the eggs are placed in this insect host. And what they're doing is these viruses are carrying genes that are wasp genes to the host larvae that downregulates the ability of the host larvae to mount an immune response. And ultimately, this allows this gruesome set of pictures where this poor caterpillar is essentially eaten alive by these larvae that end up of the wasp, but end up emerging from it. And it's only possible because of this relationship they have with pallidnaviruses. So here the punchline is unique pallidnavirus to unique wasp species allows these wasps to be highly successful biologically. And it must be that the relationship between these viruses and the wasps are quite ancient. Now, could one capitalize on systems like this where, imagine we were very worried about caterpillars and we want to release a bunch of these wasps to kill them? What we could be relying on essentially is that the virus within the wasp is doing us a favor. And that would be an enemy of my enemy as my friend. So this is what we're trying to capitalize on in virotherapy. I work on evolutionary medicine, and that's bringing evolution thinking to medical problems. And virotherapy could go like this. Here on the left, you could sample for viruses in some environment, just looking in the virus biodiversity on the planet. In this case, is an individual from my lab, Ben Chan, who is looking for bacteriophages in this natural environment. And you could bring them in the lab, characterize them, and you might be so convinced that they would be useful in medicine that you could do what he's doing here on the right. He's now in a hospital room in Texas showing a young woman who's undergoing lung failure because there is a chronic bacterial infection in her lungs that is resistant to all antibiotics that are allowable in this country. And what he's showing her is a vial of these phages that she's going to inhale over the course of 10 days with the hope that they would kill the bacteria in her lungs that antibiotics cannot, as her pediatric doctor is looking on. In her case, she has cystic fibrosis. So this disease makes her lungs very vulnerable to bacterial infections in the first place. And individuals like her are more worried than most about a problem I'm going to talk about in a moment, the antibiotic resistance crisis. That's because she's constantly battling bacteria coming into her lungs, and increasingly those bacteria are antibiotic resistant. These sobering projection maps tell us this story that by 2050, especially in the most populated parts of the world, but essentially throughout the world, you've got millions of people who are expected to be dying from antibiotic resistant bacteria. So much so that it's projected to be worse than cancer deaths annually. This is going to be a leading cause of death in humans, and that's not good news. What you can do about it, if chemical antibiotics won't work, could you turn to an old idea? And that's what we're trying to do. Phage therapy is an old idea, and it works by finding a phage that is specific to a bacterium, and it is able to kill that bacterial cell while making copies of itself. So not only is it specific to its target, but it makes copies of itself as a drug. It's got self-amplification. That's very attractive as a benefit. It's got costs, though. If it's too limited in its specificity, then it's not very useful as a broad spectrum drug. It might be difficult to deliver it to the infection site where the bacteria are causing the problem. Ultimately, you might mount an immune response to these foreign invaders coming in to try to help you, and you'll start attacking them with your own immune system. So therefore, the phage could be a target of your immunity, even though you don't really mount an immune response that's appreciable to phages that you see when you accidentally ingest them through food, water, and just breathing in dust particles in the air. But I'm an evolutionary biologist, and I predicted that if you use this, it's going to fail eventually because bacteria evolve resistance to stuff, including phages. So this is where we're trying to capitalize on our knowledge of evolution to turn that cost into a benefit. And if you work on evolutionary biology, you quickly see that it involves compromises. So selection, as described by Darwin, it's an ability to improve a trait to make it an adaptation, but it sometimes reduces performance in other traits. One thing that humans do better, presumably, than all other animals is that we throw objects with accuracy at high speed, like in professional baseball players and softball players. And this chimp is our closest relative and has a very different upper musculature to its arms because it's evolved and adapted to living in trees, or at least can live in trees. So it can hang from one arm from this tree limb for an extended period of time, even though it's a young chimp. Humans can't do that. And chimps cannot throw objects with accuracy at high speed. Another thing that's within our species as a trade-off, is an evolutionary trade-off, is that we walk around on 2 feet all the time, and there's only a great ape that does this. And that's great for us, but unfortunately it gives us lower back pain often and neck pain as a result. So the idea is that you can have evolution of some traits, but it can leave you vulnerable to other problems. And that's what we want to try with phage therapy. If you can think about it, if you find a phage that kills a bacterium, that's classic phage therapy. If you find a phage that kills a bacterium and it's able to take the evolution of that bacterium to a better place biomedically, then you've got a better phage for therapy. In this cartoon, it's a little busy, but the red phage and the purple phage are interacting with two different things in this bacterial target. The red phage is interacting with these efflux pumps, and it's hypothetically here attaching to the outermost protein. And we care about efflux pumps because if antibiotics get into the cell, they're pumped out with efflux pumps. It makes them broadly resistant to antibiotics. So you're going to use this phage and kill the cell. And to deal with the evolution of resistance to the phage, they might compromise or just entirely get rid of the efflux pumps. And that makes them antibiotic sensitive instead of resistant. So you sort of solve the problem and you set up the system so that it's even more vulnerable to something else that's already medically approved that's not currently working, and it makes it useful again. So more quickly, you can think of this purple shield around the bacteria as a capsule, and sometimes that allows them to hide detection from the immune system. And if you have a phage that's specific to the capsule, then the bacteria have to deal with the problem, and they'll evolve resistance to the phage, lose the capsule, and suddenly they become less virulent because they're detectable. So our expectation is we go out in nature, we can find these anywhere, and it depends on what the mechanism here in this hypothetical bacterial cell to determine what is the best phage to solve the problem. We hunt for phages in pristine environments, less pristine ones like wastewater treatment plants or farms, and they're very easily found. We bring them in the lab and we fully sequence them. And this kind of funny cartoon is showing in a collection recently of 100 or so phages. We can sequence them and know their genetic relatedness through these trees, and we can also put them into known phage families. So this allows us in this one case of a bunch of phages that are specific to this pathogen, Pseudomonas aeruginosa, what are these phages genetically, and how do they relate to one another? But here's something else we'd like to know, is shown by Ben Chan and my group. If you have these phages that you take from nature here lined up along the left, and we know their relatedness to one another, and you challenge them to infect, this is, I don't know, 40 or 50 or so clinical strains of a bacterium, and you know the sequences that describe how these bacteria are related to one another, we would like to now fill in this matrix, and the dark squares are showing where the phage is successfully infecting that clinical strain in this challenge assay. So immediately what you see is a lot of dark blue. And what that suggests is that phages are not so specific to their targets. You can easily find phages that actually kill the majority of these clinical strains of a bacterium they've never seen before. So it's kind of a fallacy to think that phages are too specific to be useful in therapy. But which are you going to choose as the best candidates for therapy? Should you use cocktails that bundle together the ones that are specialized in killing different of these strains, or do you just want to find one that's the most generalized and kills a lot of them on its own? So that's kind of a big question in phage therapy now. If I show you now the full data set, I know you can't make out the details, but it's hard for a human to make sense of this pattern and say in a future world, if a physician has a bacterial strain infecting their patient and they know what the sequence is of that strain, can they immediately predict which phage could kill that bacterium? We have to now take AI or machine learning to sort through these data to make that prediction. That's what we're trying to do. And ultimately, I showed you these first two cartoon depictions of trade-offs. This is from a recent paper we published that reviews this. And I already described how efflux pumps and capsules would work. But here on the right, if a bacterium has these very abundant lipopolysaccharides surrounding the cell to give it integrity, then phages could be binding to portions of that LPS in order to get in. And that's going to lead to the bacteria evolving resistance to the phage by trimming down that LPS from the cell surface and making it shorter and kind of eliminating some of the binding targets. The problem for the bacteria then is that if they trim down the LPS, it's an easier route for the antibiotic to get in, and they can become antibiotic sensitive. So now we've got three different mechanisms here that lead to greater antibiotic sensitivity. And we've had data in the laboratory. This is work by one of my postdocs, Alita Burmeister. This diagram is of an efflux pump in E. coli, and we found phages that bind to this outermost protein. And in the laboratory, if you grow them in the absence of an antibiotic, in the wild type E. coli and a genetically engineered version of it where you remove the Tol-C protein, it grows just fine, like the wild type, and a phage-resistant mutant also grows fine in the absence of antibiotic. But as soon as you grow them in the presence of tetracycline, now you find that the Tol-C knockout and this phage-resistant mutant are growing poorly. They've become antibiotic sensitive. Similarly, there's another problem that this phage causes for E. coli in that it binds to a portion of LPS. So here's a side view of LPS, and the color portions here are the portions where the phage is binding. So if you knock out those genes, and sorry, I misspoke. If the bacteria mutate at those sites, then they can gain resistance to the phage, but now they have a different problem. They become sensitive to colistin, which is an antibiotic, because they've trimmed down their LPS too much. So everything that I just said there in the second portion is that we're finding phages that are useful in therapy. We're characterizing them enough that we have confidence that we can put them in people, but immediately there's a lot of issues that might prevent them from working in a person, because you're kind of taking lab data immediately to the black box of the human body and trying to figure out if it can work. All kinds of things could stand in the way. Would phage therapy affect interactions with your cells for some reason? Other members of your microbiome, even phages that are there all the time, other bacteria that are there, other pathogens might get an upper hand if you take a phage and remove one pathogen. Is it actually making it easier for another pathogen to come in? So these are all kind of questions we grapple with, and yet we move forward. So this is a very busy summary of the first person we treated with a phage, because he had run out of other options, was an elderly man who had an aortic arch replacement. I think you heard about him or read about him in the Scientific American article, so I won't go at this at length. But this is the stuff that was remarkable even to us, is that one, we can find volunteers who have no options, who are willing to undergo experimental therapy, and two, it actually works when you deploy it. I did not think we would be so successful, and here are the data that we haven't even published yet, where in this case, most of this table, the first row is the man who you read about or heard about, and the others are a bunch of other patients who all have a similar problem. They have a lung architecture that makes them vulnerable to bacterial infections, and those are increasingly antibiotic resistant. So those who are have the genetic disease of CF, or if they are just suffering with bronchiectasis or chronic obstructive pulmonary disease, then they also have these problems of bacteria coming in and causing lung failure and sometimes death. And if the bacteria are resistant to antibiotics, then they've got a big problem. So these are exactly the kind of folks we've been helping. How we help them is we administer the therapy through inhalation. They simply inhale the phages over the course of several days. We do this, of course, in collaboration with doctors. So John and Gail here are the doctors who we've been working with most recently. And we are also capable of taking from these patients sputum samples so they can cough up the mucus from their lungs. And we can look in that mucus for the detection of microbes to see whether this is working. But the first thing we can do is measure, is their lung function improving? And here in red, after therapy, yes, it improves. And the bacterial load as detected in their sputum is going down during phage therapy. Further good news. And here's a summary of different antibiotics. And in many of the cases, the greater sensitivity of antibiotics is detected in the bacteria that we take from their sputum. So in other words, it's mimicking what we see in the laboratory. Now this inflammatory molecule reduction, I have to spend another slide to explain that. So some phages are specific to pili, this pilus of a bacterial cell. And in the case of P. aeruginosa, the pili are associated with a virulence factor called a chemical called piocyanin. So these phages that we have found bind to the pili, and they use that to enter and kill the bacteria. And this selects for the bacteria to get rid of the pili. And when they do that, they are debilitated in their ability to make piocyanin, which is this nasty chemical which glows bright green, and it allows the bacteria to be an effective pathogen that causes a bunch of tissue inflammation and destruction in the human lung. So what we found is that the patients who receive this phage, indeed their bacteria are removed from the lungs, but when we find in their sputum the remaining bacteria, they trough out here in their ability to make piocyanin. So they are greatly reduced in their ability to make piocyanin. If they're still in the human body, we don't really care about them. And this is a little busy. I'll just say it quickly. If you have questions about it, I can answer it. But these are different inflammatory markers. So you can kind of take the bacteria from their sputum, grow it up in the lab, throw out the cells, but you take the supernatant and you put it on airway epithelial cells to see whether it's causing stress in cells of human origin. And we find that after therapy, there's much less stress that these cells are undergoing. They don't undergo inflammation and mount an immune response because the bacteria are not hurting them as much. So the next steps are kind of obvious. We have an FDA approval to do a clinical trial to study whether phages would protect healthy volunteers who have cystic fibrosis. If you give them phages over many months, would they be better protected against these bacteria coming in and causing them harm? We also teach this technology to undergraduates here at Yale. We also teach it in workshops across the world, but especially in countries where people might be relying more on phage therapy in the future because it's a very inexpensive option. So this is a recent workshop we held in West Africa. And the next steps also involve non-human systems. Antibiotic resistance is a big problem in humans, but it's also a big problem in plant and animal agriculture and in aquaculture, where this stuff can economically collapse when you have antibiotic resistant bacteria coming in and causing devastation. So therefore it threatens food security. So we're trying to work against that. And the last would be zoo and aquarium animals are quite precious to zoo and aquaria because they can't really take them out of nature anymore. And already folks in my group have worked with veterinarians in Florida for a sea turtle that was suffering a multi-drug resistant bacterial infection. And we hunted for phages that would kill those bacteria as well as exert selection pressure for trade-offs so that the bacteria could be more easily killed with antibiotics. So I hope you've enjoyed what I've presented. Viruses are far from trivial, but I'm going to end with a trivia question because I love trivia questions. So think about the following. And this does involve viruses, believe it or not. So what would you trade for 4 tons of wheat, 8 tons of rye, 4 large oxen, 8 large pigs, 12 large sheep, 2 hogshead of wine, 4 barrels of beer, 2 barrels of butter, 1,000 pounds of cheese, 1 bed, 1 full dress suit, and 1 silver goblet? All of this was traded for exactly 1 item. Was it a vaccine, a flower, a ship, a horse, or a royal family member? And I can't see you all. I will give you exactly 5 seconds to think of the answers as we're running out of time. Anybody want to hazard an answer? Nobody knows.
Looks like there are some guesses in the chat.
Oh, really? Would you mind looking at those for me?
Lisa says flower. Tracy says A.
Okay.
Kieran says smallpox vaccine.
Huh.
Couple of A's. Lots of A's. Couple of E's.
Well, everybody has vaccines on their mind. Fortunately, you're wrong. Flower. So at the height of the tulip craze in Holland in 1625, all those goods were traded for a single bulb of what's called a broken colored tulip. But that is trivial to create now because that plant has been infected with a tulip breaking virus that breaks up the color scheme of the flower. And back then it was such a precious commodity that people would trade an amazing amount of stuff for it. And now it would be literally trivial to create it by infecting the plant purposefully with the virus. All right, so I left, I've already gone over, but I can hang out a little bit if you can, like a few more minutes to answer questions. And wow,
I still think vaccine is worth it. I think
Vaccine
I
is
still
worth
think
it. I tell you
vaccine
what,
is.
I agree. I would trade all that if I owned it for a vaccine that would work.
Well, thank you so much. Yeah, Paul, if you don't mind answering 1 or 2 questions, there were some in the chat. Maybe Sibylla, I think
I'll let you can choose for me.
Okay, sure. Yeah, Sibylla, I think you had a couple of questions. Would you like to pose one now?
Yeah, I found it interesting that those Politna viruses that the wasp kind of uses to her advantage. I was wondering what the definition is. When does one say it's a virus, and when could one classify it as part of the organism? Like if you think of the endosymbiotic hypothesis with mitochondria, you could say like at one time maybe it was like the viruses, and in the future could that be an essential part of that wasp?
Right. Good question. So basically what's going on in these wasps, they deliver something that, through microscopy looks just like a virus particle, except there's no virus genes in it, right? So that suggests, and I take your question, I mean, these viruses are kind of suckers, right? They've been taken advantage of and co-opted to do something. However, that tends to be the popular notion, and I'm trying to push back on that a little bit because I think selection takes systems down some paths that to us may seem like, oh, that's a weird path to travel, but the loss of autonomy, giving up all of it to your host. You host, do with me what you will, as long as you give me a cozy environment to replicate in. It starts to create this fine line between what is the individual? Is the virus no longer a virus? Is it completely a sucker that's been co-opted? Or is it like, hey, I trust you, host. Do with me what you will, and I'll follow your path. And maybe that's a typical evolutionary endpoint for some viruses as a strategy to evolve. I think we're a little naive sometimes when we think about what the options are in biological systems, but that was a terrific question.
Okay, and then maybe one more question. Kaylin, I think you had put one on phages in the chat as well.
Yeah, I was just wondering if there's kind of like a database that's being collected, because I'm sure you would be accepting them from all over, anywhere you can get them, right?
Mm-hmm. The
Any may be applicable.
Yeah, so this is the tricky thing, especially UC San Diego. They've had a lot of success also with phage therapy, and they're more enamored of building a huge library of phages that are fully sequenced and kind of a little bit characterized, making them available to physicians. And we're a little bit more to the side of, okay, I'm going to look for a lot of phages, but I'm going to whittle it down to the most interesting ones and really characterize the heck out of those. And I would rather give to physicians like one of 5 or 6 viruses that I think are trusted enough and known enough that are going to do the job and not create surprises. So there's a little bit of these different philosophies about how do you look through the vastness of the biodiversity of phages and find good candidates. Some people are like free for all, and others like me are like, I'd rather work on a few and know more about them because we're already putting them in people based on the best evidence we have. But it takes a lot of time and energy to figure out biological details, right? We don't want surprises. And I feel like I'm dealing with 5, 6 phages. I might be surprised by them, but that's a lot easier than if I'm dealing with 200 of them and deploying those in people. So it's a little unclear now, but the popularity of this is growing. There's a phage center in Mayo Clinic. There's a phage center in Texas A&M, UC San Diego. We essentially have one at Yale, but it's kind of my lab and the med school talking to one another. We have a phage center of sorts, and we're going to try and give it a specific name so that people would actually recognize Yale. Yale has a center, but it's a little unclear the path right now. Phage biotechnology is extremely popular. At the time when Rebecca was working with me in my laboratory, it was kind of hard to get phage work even supported from a grant. And now people are using phages for all kinds of things, alternative energy sources, different batteries could be made using phages, this kind of work on viral therapy. It's a bit endless right now with what people are excited to do with phages through biotechnology approaches in solving human problems. So it's a really good indicator of, yes, viruses can stop the world, but if you know enough about them and can trust them to solve human problems, then they can do amazing things as well.
Wow. Well, thank you so much, Paul.